In the fight against cancer, a new alliance is forming. It's one between artificial intelligence and biology. Now, these two worlds have traditionally operated very differently, but their combination is starting to open up new possibilities. So today, we're going to explore how this partnership is taking shape and what it could mean for one of our biggest medical challenges.
The central challenge with cancer is its complexity. It isn't a single point of failure. Instead, it's what's known as a network disease, a cascade of disruptions within the vast, intricate web of molecular interactions inside our cells. To make any real progress, to understand it, and eventually treat it more effectively, we need models that can grapple with this level of complexity.
Historically, researchers have approached this problem from two very different directions. One is what we call the data-driven approach, which is the world of machine learning. The other is the theory-driven approach, which is known as mechanistic modeling. Let's take a look at how these two paradigms work. All right, let's compare these two directly. Machine learning models, or ML for short, are fundamentally data-driven. You provide them with vast data sets. Think of things like genomic or proteomic data, and they excel at identifying patterns that might be invisible to a human observer. The primary limitation, though, is that they can be a black box. We bet a prediction, but we don't always understand the underlying reasoning. Now, mechanistic models are the opposite. They are theory-driven. They're built from the ground up based on established biological and physical laws. This gives us clear causal insights. Their weakness? They often have difficulty scaling to the full complexity of a biological system.
So if we look at the typical machine learning workflow, it begins with large-scale data collection. This raw data is then processed. It's cleaned, normalized, and undergoes something called feature engineering, which is the crucial step of selecting the most relevant variables for the model. From there, the model is trained on this prepared data to learn how to map inputs to specific outputs. Finally, its performance is evaluated using a separate set of testing data to see how well it generalizes. The entire process is centered on learning patterns directly from the data.
Now, let's contrast that with the mechanistic modeling workflow. This process begins not with data, but with theory, with the known biological principles that govern a system. These principles are then translated into a mathematical framework, often using things like ordinary differential equations, or ODEs, to describe the interactions. Experimental data is then used, but here it's to estimate any unknown parameters within those equations. Once the model is calibrated, it can be simulated to predict how the system will behave over time, all based on our fundamental understanding of the biology.
And this leads to a key insight, as stated by the researchers in the paper we're looking at. These two approaches possess complementary strengths and limitations. The weakness of one is often the strength of the other. So the logical next step is to ask, what happens if we combine them? This is the core idea behind the development of hybrid models.
The first level of combining these methods is known as shallow integration. You can think of this as the initial way these two distinct modeling worlds begin to interact. So what does shallow integration actually mean? Well, in this approach, the models remain structurally independent. The key is that the output of one model serves as the input for the other. For example, a mechanistic model could generate a large set of simulation data, which is then fed into a machine learning model for pattern analysis. The internal workings of each model aren't changed. They simply work in sequence.
This method has several key applications. One is for sensitivity analysis, where machine learning can be used to efficiently determine which parameters have the most significant impact on a mechanistic model's output. Another is in building surrogate models. Here, an ML model is trained to approximate the behavior of a computationally expensive mechanistic model, allowing for much faster predictions. And finally, it's used for feature engineering, where a mechanistic model can generate new, theory-informed features to enhance a limited experimental dataset before it's used to train an ML model.
The utility of surrogate models, in particular, is significant. The increase in computational speed can be dramatic. A simulation that might take a mechanistic model hours to complete can be replicated by a trained surrogate model in a matter of seconds. This kind of acceleration makes large-scale analyses that were previously impractical now feasible.
Beyond this sequential approach, however, is a more profound level of combination called deep integration. And here, we're no longer talking about two separate models working together. We're talking about a single, truly hybrid system.
In deep integration, the line between the two approaches begins to blur. Here, principles from mechanistic models, biological knowledge, are directly embedded into the architecture of a machine learning model. This creates a unified framework that is simultaneously informed by both theory and data from its very foundation.
This approach enables several advanced applications. For instance, we can design neural network architectures that mirror the structure of known biological pathways, which can make the models more interpretable. It also allows for data-driven optimization, using ML methods to more efficiently calibrate the parameters of mechanistic models. Perhaps most significantly, it opens the door to data-driven mechanism discovery, where these hybrid models can infer the structure of biological networks directly from observational data.
A prime example of deep integration is a class of models known as neural ordinary differential equations, or neural ODEs. Unlike a traditional mechanistic model, which is based on predefined equations, a neural ODE uses a neural network to learn the system's underlying dynamics, the rules governing its rate of change, directly from time series data. In essence, it can discover the governing principles of a system from observation alone, which is particularly useful when the underlying mechanisms are not fully understood.
Now that we've outlined these methods of integration, we can turn to the broader outlook for this field, assessing both its potential and the challenges that remain. This visual provides a good summary of the landscape. On one end of the spectrum, we have purely mechanistic models, which are high in theory but can be limited by data. On the other end, we have pure machine learning models, which are high in data dependency but low in built-in theory. Hybrid models occupy the crucial middle ground, aiming to balance these two by leveraging biological theory to better interpret and utilize available data.
The advantages of this integration are becoming increasingly evident. They include improved computational efficiency and enhanced prediction accuracy, particularly in scenarios with limited data. Importantly, by incorporating known biological constraints, these hybrid models tend to be more interpretable than their pure machine learning counterparts, moving away from the black box problem and providing more mechanistic insight.
However, significant challenges do remain. Issues of data quality, standardization, and handling missing information are persistent. The computational scalability required to handle complex multiomics datasets is a major technical hurdle. Furthermore, developing rigorous frameworks for validating these novel hybrid models is critical. And finally, progress relies heavily on effective interdisciplinary collaboration between computational scientists, biologists, and clinicians.
This leads us to the central question that motivates this entire field of research. The fusion of biological theory with artificial intelligence presents an opportunity to design a future where cancer treatment might not only be personalized, but also predictable. The ultimate goal is to shift from reactive personalization based on existing data to a proactive, predictive model of medicine, one that could anticipate the course of a disease and inform therapeutic strategies in advance.